So I'm going to begin with talking about the biological problem domain, which is one that we haven't really heard much about. And then I'll tell you about a new algorithm that we've written so that we can actually solve some problems in this domain. I'll only talk a little bit about that, but Spatially has a poster on that. So if you want more information about the algorithm, you can go see the poster. And then I'll tell you some results of our simulation experiments, though the results actually were run with the older version of the algorithm, but we've begun using the new version. Okay, so I'm interested in learning and synaptic plasticity. Now, learning is really a behavioral phenomenon. It's a change in some behavior, okay? It's a change in some behavior due to some experience. Whereas long-term synaptic plasticity is really a long-lasting change in the strength of connections between brain cells, and it's due to repeated activation of the neuron by specific patterns. So after potentiation, the neuron will actually fire more strongly in response to its input. Now, sort of the persistence, it lasts for an hour or more of this phenomenon, and the fact that it's activity dependent and it's a change, so that sounds very much like learning. So the neuron is learning by changing its activity in response to some training paradigm. The most commonly studied form of synaptic plasticity is in the hippocampal. It's hippocampal long-term potentiation. And so I'm going to be talking about some results of these experiments and how we're trying to model them to understand some of the results. So I just wanted to show you the setup. These are tiny little brain slices from a mouse hippocampus, which sort of looks like this. And what we have is we have a recording electrode. We're recording population responses from outside the neurons, and that's with this pipette. And then we're going to stimulate some axon fibers, and that's with this pipette. And what we see is this black response, roughly a couple millivolt response to stimulation. We can record that for a long time and it stays stable. But then if we give some sort of higher frequency stimulation, what we'll see afterwards is a response like this. And so in the other graphs, I'm just going to be reporting this is the stimulation artifact. So I'll be reporting the amplitude between this and this peak point right here. And so that's shown here. If we give one train of 100 hertz, such as 100 pulses for 1 second, we'll get this increase in the amplitude, and then it will slowly decay, and if we keep recording, it will eventually reach this basal level. However, if we give 4 trains, we get this, the amplitude isn't really bigger, but it's much longer lasting, and in fact, it will stay elevated pretty much for as long as we can record it for. Thankfully, we don't have to do simulations for multiple hours because the induction of this persistence happens right around here. So during the 20 minutes, nonetheless, we do need to simulate events for around 20 minutes or so. Okay, so what are some of the molecules involved? Well, probably the most well-known is a kinase called calcium calmodulin-dependent protein kinase, type 2. But in addition, there are many other molecules and kinases. One in particular is protein kinase A. And what this graph shows is if we have some mutant mice which have a deficit in their protein kinase A, and in this case we're doing a behavior where they're learning in this big tub, they're learning that in one of the quadrants is an escape platform. And these mice don't like being wet, so they'll swim to the platform. And you train them to learn where the platform is, and then you test them later. And what this is showing, this is the wild-type animal, which is spending about 50% of its time in the quadrant where the platform is. What you've done is you've removed the platform. These poor animals are just swimming around saying, I know it was here just a short time ago, sort of like, I know I left my keys in the bedroom. In any event, if in these protein kinase A mutants, you see it's spending much less time in the quadrant where the platform was. This is another task, which is contextual fear conditioning. So you put an animal in a different type of box from its home cage, and then you give it a tone, and then a foot shock, and what you see is the animal, even 24 hours later, will freeze when you put it into that box. It's learned to associate the box with this fearful event. Again, these mutants will show the freezing immediately. That's one of the behaviors that a mouse will do when it's scared. It will show the freezing an hour later, but a day later, these mutants are showing some deficit. Okay, so one of the first questions I started addressing was why does the requirement for protein kinase A in the synaptic plasticity depend on the temporal interval of stimulation? So what do I mean by that? Well, here, the blue is the wild type response. So if we give 4 100 hertz trains, shown by these arrows, and they're 5 minutes apart, I get this long-lasting LTP. If I give these same stimuli, the same 4 trains, but now they're separated by 3 seconds, I get this long-lasting LTP. But these PKA mutants have fine LTP in these mutants with 3 seconds. But if it's 5 minutes apart, I see an LTP deficit. Again, I'm talking about the long-lasting phase, so I'm not talking about this initial response. I'm talking about this response right here. So why do I see that deficit? So I started with just a simple single compartment deterministic model of these signaling pathways. And what I saw is that both protein kinase A and CAM kinase have opposite temporal sensitivities. Namely, the protein kinase A increased as you increase the intertrain interval, whereas the CAM kinase decreased as you increase the intertrain interval. And so we predicted that somewhere around here, you would, at this interval, you would switch from a PKA dependent to a PKA independent paradigm. And so again, that interval is 60 seconds. We made that prediction. My collaborator said, okay, so we'll do the experiments. You want 55 and 65 seconds? And I said, no, I'm not that convinced I'm so precise. So we did 40 seconds. Indeed, it is independent of protein kinase A. The blue is the control. The red is with a protein kinase A inhibitor. And then we did 80 seconds. And again, blue is control. And the red shows this deficit with a protein kinase A inhibitor. So we were really happy about that. But of course, this is a conference on spatial stochastic simulations. And so what's awesome is I don't have to explain to you guys why we should not be doing this in the single compartment model, why noise matters, why space matters. So we're just going to move on. But that was my first experimental prediction that was correct, yes.
In the single compartment model, there is another attractor, and you get to the basin of the other attractor?
I didn't analyze that way. We actually did these simulations in XPP, but we have about 50 or more differential equations, and it's actually, and we don't, it's very difficult.
If you run the top one forever, would it stay there or would it come down?
So this is not the dynamics part. I'm plotting for you the area under the curve of the response, okay?
Can you explain a bit the component of the simulation? Because usually you have to start with the calcium. Calcium has to bind the calmodulin. Can you explain a little bit?
I will.
Okay.
I'll do that when I get to the multi-compartment. Is that okay? All right. So again, back to some of the experiments that I didn't do, but that are motivating the modeling I'm doing, is that the molecular requirements are actually quite diverse. It's not just, yes, it is calcium, but it's not just calcium and CAM kinase and protein kinase A. So one of the observations is that beta adrenergic receptor stimulation is sometimes required. So if you give one train of 100 hertz, but in addition you bath apply, say, norepinephrine or a norepinephrine-like drug, you will go from this short-lasting LTP to a much longer, to this late phase, which lasts pretty much forever. And this is indeed dependent on protein kinase A, because if you inhibit the protein kinase A, what you see is it starts coming down, and so it's much lower. There's another type, which is we can give a 5 Hz stimulation, and that's shown right here for 3 minutes, and it really doesn't produce anything except this transient depression. But again, if we add the isoproterenol, I'm sorry, here this is not pKa dependent. These two are the same. So this requires beta adrenergic receptor stimulation, not pKa dependent. But if instead of 1 Hz plus isoproterenol, we give 5 Hz plus isoproterenol, we get the same long-lasting LTP, and now we are pKa dependent. We see this big difference. Okay, so again, why is that? Why do we have this difference? It's different temporal patterns have different requirements. So I would say that one of the most important unsolved problems in neuroscience is how can all these different forms of this long-lasting LTP depend on different molecules? Or put another way, why do we need so many different kinases and other molecules for the induction of long-lasting LTP and memory? And in particular, I'm going to try to address what is the role of the beta adrenergic receptor. And just keep in mind that the induction of the process occurs within 10 to 20 minutes. And what I mean by that is there are other experiments showing you can add inhibitors only during the 10 or 20 minutes surrounding the induction, and then you wash them out and you don't see the deficit right away, you see the deficit a long time later. All right, so of course we're going to create this computational model, and it will be spatial. But before I explain about the model, clearly there are very diverse time scales and spatial scales of the molecule activation. And so this is some research by others with some imaging showing this gray is the time scale of the calcium pulses, whereas the CAM kinase goes up much more slowly, and this is seconds. But now we could image other molecules, and some of them come back down to basal within a couple minutes. Some of them are activated for a long time. And this is showing different spatial scales. So what they're doing is they're imaging RAS. They're activating just the spine. They're imaging Rho A. They're imaging CDC42. These are 3 molecules that are involved in actin dynamics. And now they're plotting the diffusion of the activity along the dendrite. And what we can see is that the spatial scale of these molecules is quite different. So some of them spread over, tens of microns, and some of them remain restricted to the spine. But we don't see a bimodal distribution of time scales or spatial scales or trimodal. There's really a continuum. And furthermore, some molecules have a very high propensity early, and then we'll have a low propensity. So whether you wanted, if you want to say do some molecules one way and some the other, you can't really partition cleanly, and furthermore you have to repartition. Okay, so we created a new simulator, which is indeed a hybrid of tau leaping and an exact stochastic, but and so we do indeed subdivide our morphology into voxels. But we're not going to do an explicit partitioning of the space. We do have both reaction events and diffusion events, and we will indeed schedule a single event if the population, the diffusion population, or the reaction rate, i.e. if the propensity is small, and then we'll schedule multiple events if the population or the reaction rate is large. But again, there's no explicit partitioning. So as each event comes up and is scheduled, it will be scheduled appropriately. So again, we're going to do what others have done before and use a priority queue. So in this very simple reaction where we have 2 voxels and a single reaction, the diffusion of B from voxel 0 to 1 might be at the top of the queue, and then we have another reaction which are subsequent to that. So this is the high priority, this is the low priority. So again, we're going to order the reactions, including diffusion, by the time of the next event, but we'll either schedule a leap or a single event, depending on which is more efficient. Okay. So all I'm going to say about the algorithm, again, you can get more information about that later. So here's just one example where we compared accuracy. We did, again, a fairly simple a plus b goes to null. It's a one-dimensional domain of length 40. So here's our 100 voxels, and we're initializing some molecule in the lower left corner of B, and we have 1,000 A distributed, and so this is the time, and this is if we simulate it exactly, and this if we do what we call our asynchronous tau leaping. The other thing we do is with this leaping, we don't leap all events at once. We leap the single event at the top of the queue, and then the next event comes up. And so that allows us to avoid some of the issues of depleting molecule numbers by leaping all the events at once. Okay, so then we used a much larger model, which was dopamine-dependent production of cyclic AMP, activating protein kinase A, which then phosphorylated phosphodiesterases, which reduced the cyclic AMP. So really just a fraction of the model that I'll be showing you later. And then we would implement that in either 3 or 6 or 9 voxels, so we would scale this model. And what we see, the blue is the runtime of our previous pretty fast fixed time step tau leaping, and now this is the runtime of our new algorithm, and this is a logarithmic axis. This is it as we increase the size of the model, and this is either the number of events or the runtime, and it scales fairly linearly in this log space. And so we're basically getting a 3-fold increase in speed. So simulations that used to take us 3 days, we're now doing in 1 day, which is for, say, I think around 500 or 600 seconds of simulation time we can do in 1 day in our models. And the other thing, I'm not going to talk, I don't have simulations for you, but if we want to do most of the simulations I'll show you is a 2-dimensional morphology, such as this, where we have diffusion in 2 dimensions, and we say have submembrane or surface voxels in cytosolic, and we haven't yet done 3-dimensional ones, but we can do this sort of cylindrical type meshing so that we can maintain our computational efficiency while still capturing the key shape properties of dendrites. And we can also add on these protuberances called dendritic spines, and we can define different shapes and then distribute them according to densities that have been measured. And then the last thing I want to say is that, so we use an XML interface, and we actually have these separate files, and we do this because I like to emphasize with my students and postdocs good experimental design for our simulations, because we're really trying to address biological questions. And so we have separate reaction files and the morphology and an initial conditions file, and I think of this as the tissue, and then we have a stimulation file, and we specify our output. And this is really the experiment, though there's some overlap, but this allows you to say, well, how does the morphology affect what I'm doing, and then you know you've maintained the same reactions because you're using the same file. Or how does in this case, the frequency of my stimulation, sorry, that would be the stimulation file, how does the frequency of my stimulation affect, my output, my kinase activation, and it's just much easier to keep track of. Okay, so back to the biological question, which is I'll rephrase it as, can a single molecular signature predict the induction of this long-lasting form of LTP? So this is the model we're using. Indeed, we have calcium influx. We don't actually have the NMDA receptors. We are actually injecting calcium, which can bind to calmodulin, becoming this activated complex. The calcium calmodulin can bind to either a phosphodiesterase or a phosphatase or this calcium calmodulin-dependent protein kinase. We have a pool of calmodulin that's actually bound to neurogranin. We have this beta-adrenergic receptor, which when activated leads to GS production, which activates adenylate cyclase. The adenylate cyclase is synergistically activated by GS and calmodulin, and when active it produces cyclic AMP, which then binds to either protein kinase A or EPAC, another molecule that's been implicated in synaptic plasticity, some forms. PKA can actually phosphorylate the phosphodiesterase, one of the phosphodiesterases that then enhances its inhibition. The other thing we have in this model, which is pretty unique among the synaptic plasticity models, is something called GS to GI switching, which I'll talk about a little bit later. But essentially, if the beta-adrenergic receptor is stimulated for too long, it actually uncouples from the GS type of GTP binding protein and can now activate GI proteins and lead to ERK recruitment. So we have the GI recruitment. We haven't yet added ERK to our model, though. And we have a couple of readouts of the PKA, and one is this inhibitor 1. We can also look at phosphodiesterase phosphorylation. In a previous model, we've had the AMPA, and we also have the AMPA receptors. I'm sorry, we have AMPA receptors, which then can be phosphorylated by either the CAM kinase or the protein kinase A, and also can be dephosphorylated. Okay, so most of the simulations I'm going to talk about are done in this very short 2-micron dendritic segment with a single spine. In the spine, there's axial diffusion, calcium influx at this postsynaptic density region, and also into the dendrite to, as would occur with activation of voltage-gated channels. A few of the simulations use this 20-micron long morphology with multiple spines. And our stimulation patterns will indeed be patterns of calcium influx and a beta-adrenergic ligand. So here we have some of the different stimulation patterns. We can give one train of 1 second of stimulation. This would be activation. This would be pulses of calcium influx and brief pulses of beta-adrenergic agonist. Or we can give one train and a bath-applied beta-adrenergic agonist. This is the outcome. This is an early phase of LTP. This is a late phase. Or we can give just this low-frequency pulses of calcium. That produces no change by itself, but we compare this with isoproterenol. We get the late phase of LTP. And now we have also our 4 trains of high-frequency stimulation at 80 seconds also produces a late phase of LTP. And then 2 other paradigms we use are 4 seconds of high-frequency stimulation only separated 3 seconds, or isoproterenol alone. And we're going to also validate the model by repeating many or all of these protocols with protein kinase A inhibited. Okay, so what we discovered with lots of simulations and analysis of the molecule outputs is that we need 2 molecular signatures. So what we're going to do is add up the phosphorylated CAM kinase, the activated EPAC, the activated PKA in the spine. And we'll also do the same thing in the dendrite. Though in the dendrite, we're going to separate out the PKA into 2 terms. One is the phosphorylation of this inhibitor 1 and phosphodiesterase, and the other is this switching term. And to produce LTP, both of the signatures have to cross this threshold for a minimum duration. And so this is showing the signatures for this 4 trains of high frequency. In black would be separated by 80 seconds, and you can see the signature goes up and down. And by the way, we don't have a single precise signature. This is showing you a range of signatures that we're using, and any value in between the upper and the lower green dashed line will give us the same result. Okay, if we give 3 seconds of stimulation, then we can't see the distinct trains, and we just see one large increase, which then decays. And so we're simulating this for 900 seconds. This is the spine signature, and this is the dendritic signature. And you can see that for both of these cases, both signatures cross the threshold. And so now this shows that indeed with this model we need this beta-adrenergic stimulation when we give this weak or a single train of 100 hertz. So again, this up here is the spine signature without the isoproterenol. Down here is the dendritic signature. In this case, nothing is crossing the threshold, neither the high frequency train or the low frequency train. Up here, we do cross the threshold for the high frequency train, but not the low frequency. But neither of these will give us LTP, but now we add in isoproternal, and that shows when we added it in, and now the isoproternal alone will cause an increase in the dendritic signature, but not the spine signature. But both the isoproternal plus high frequency and iso plus low frequency, both dendritic and spine signatures cross the threshold. Okay, so now we'll simulate the case of pKa inhibition. And again, the pKa inhibition blocks LLTP for the appropriate paradigms. So again, here's the signature in the spine, the signature in the dendrite. The isoproternal plus HFS is in red. Both signatures still cross the threshold as they should. 4 trains of high frequency stimulation for 3 seconds. Both signatures cross the threshold. Both of these paradigms are not blocked by pKa inhibitors, but these other 2 paradigms, indeed, their signature is below the threshold, one or the other. So now we want to ask, what is the role of the beta adrenergic receptor? And as I mentioned, here's the if you back apply isoproternal, the beta-2 receptor, which normally is coupled to the GS signaling, will now become phosphorylated by pKa. Now it couples to GI, which indirectly recruits ERK. And here's some of the experimental work which basically supports this switching pathway. This is discovered by Lefkowitz. What he added was a different beta adrenergic agonist. And what you see is that produced a large amount of phosphorylated MAP kinase. But if you add this pKa inhibitor, you're going to decrease the amount of phosphomap kinase. Here's another graph which shows that the GI inhibitor, right here, pertussis toxin, is also blocking to the same degree the recruitment of MAP kinase. So it's the GI inhibition, therefore it's not because it's working through protein kinase A. And this is another graph showing that indeed some of these LTP paradigms require ERK, so that if you inhibit ERK, you don't get the LTP. Okay, so we want to know one of the questions is the beta receptor required for all forms of LTP? And most people have said, no, it's not. If you just give electrical stimulation, the beta receptor is not required, because when you add propranolol, which is an inhibitor of the beta receptor, you do not block this 4 trains of the LTP produced by 4 trains of high frequency stimulation. However, this is other research by Kasai and others, which shows that when you add propranolol, indeed it blocks the cyclic AMP, but it does not block the ERK activation. Here's this other drug, ICI 118551, actually is a complete antagonist. And so this shows that it blocks a different type of LTP that I haven't talked about, but a theta burst. So here's the vehicle control, and here's the ICI produces this decrease. And this is this theta burst is another electrical form of LTP that does not require bath-applied isoproternal. Okay, so now what we wanted to do is repeat these simulations with either propranolol or this ICI, and here's our control without the drugs. And what we see is that when we add the propranolol, it indeed reduces the dendritic signature, but it still crosses the threshold, whereas ICI reduces it a little bit more, and it no longer crosses the threshold. So this would be our prediction that the ICI would block 4-train LTP, and that is being tested by our collaborator. So I want to talk a little bit more about spatial properties of LTP. Again, this is our neuron actually looks more like this. Here's the cell body, a single cell body, which has many dendrites, and maybe only parts of the tips of the dendrite or other parts of the dendrite is going to be active during any LTP induction paradigm. And so one question is is how we achieve this spatial specificity. In particular, this late phase LTP requires protein synthesis and transcription. Well, that happens in the cell body. So how in the world can we only reinforce those spines that were stimulated? And we're not even positive that we do only reinforce the single spines that are stimulated. It's possible that we're reinforcing nearby spines. So another question is just how much spatial specificity do we achieve? And in addition, how does the different spatial pattern of activation influence synaptic plasticity? And this is important because the spatial pattern actually represents specific environmental stimuli. So a yellow flower will produce a different spatial pattern of activity on different neurons than, say, looking at a red house. Okay, I'm going to skip these. Okay, so we repeated our simulation, and now we've got this morphology, and we're stimulating either these two spines or this spine and I think this spine. And so one question is, are we going to see an elevation in the signature of all of our spines or just some of our spines? And does that depend on whether the two stimulated spines are adjacent to one another or non-adjacent? So this is showing that this is the time, this is the spine number, and then this is the color bar, and this is where our threshold is here in this light orange. And so when you stimulate adjacent spines, you can see that this gets to be a brighter, a warmer color than these other non-stimulated spines. And when they're non-adjacent, we see a similar pattern. And so what's nice is that we're seeing a spatial specificity here, but indeed we are seeing an elevation of the signature of these other spines. And so what's really nice is that this suggests a phenomenon called tagging, whereby there's something about the stimulated spines, right, this exceeding the threshold, which they're now in a state that they can capture synthesized proteins that may be transported down the dendrite. But what's been observed is something called a metaplasticity, a heterosynaptic metaplasticity. What that means is when they've stimulated single spines, they've then observed, and producing LLTP, and this was an experiment done by Carl Svoboda's lab, they've noticed that a nearby spine now has a lower threshold for producing synaptic plasticity. And so we might be capturing that here. And this is just a different way of analyzing the response. So if we look at the amount of time spent above the threshold for the non-stimulated spines, it's distributed here, and for the stimulated spines, it's distributed here. So showing a clear difference in the response of these two spines. We don't see that spatial specificity in the dendrite. No matter which stimulation we get, we see an elevation above the threshold all along the dendrite. Again, this is only 20 microns long, so I'm not saying we wouldn't see any spatial specificity, but at least on this scale. But this is also consistent with other experiments done where they've shown that if you indeed just stimulate two spines on a dendritic shaft, you actually see activation of the whole shaft and an ERK signal, phospho-ERK sent to the nucleus. So it's very likely that, again, even with a longer dendrite, we won't see much spatial specificity, and that's as it should be. So in conclusion, one of the things I showed you is that our dendritic signature actually looks very similar, both in the spine and the dendrite. I mean, I didn't show you, I told you what the molecules were. We're looking at protein kinase A, we're looking at EPAC, we're looking at CAM kinase. But we think they're doing different things. So there's lots of evidence that shows that all three of these molecules are involved in actin reorganization as well as phosphorylation of AMPA receptors. Whereas in the dendrite, these same three molecules are leading to ERK activation, which can then regulate protein synthesis and possibly even, again, indirectly send a signal to the nucleus for transcription. So that's actually where, expanding our model, we want to add some of these downstream events, and then really, instead of having a dendritic signature of three molecules, we might just be looking at actin reorganization or ERK activation. Okay, and last I want to conclude with thanking the people that did the work. So Asha did much of the modeling of the signaling pathways, and Spatially has been, as I said, working on a rewrite of the code. And thank my funding agencies. My collaborator Ted Abel, who does the does the experiments to test our model predictions.
Thank you. So, any questions? Yes.
I just want to say I've heard about protein kinase A before, but I didn't remember what it does. I always forget what it does.
Oh, another memory kinase, they like to say. So.
David?
So, about 10 years ago, people would say that when calcium was big enough in the dendritic spine, then LTP was somehow triggered.
Yes.
So, and what is LTP in the long range? People have shown that it was due to the increase in the number, for example, of umpire receptors, so that you get an increase in this EPSP. Now here, if I understand correctly, you have identified some kind of signature to push forward this boundary, this knowledge that we do not understand between the activation and the suddenly the increase in the number of umpire receptors. So can you explain more precisely what is this signature? What is this amount of protein that has to be activated? Is it a class? You need to have a certain number of them. You need to have a certain composition of them.
Right.
Can you explain the signature?
So the signature is basically looking at the quantity of activated protein kinase A, the quantity of the activated EPAC, and the quantity of the phosphorylated CAM kinase. And we're normalizing it from across its full range that we observe and adding them together. It's fairly ad hoc. Okay, we didn't need to look at anything more complicated. This is a stepping stone for us. We're well aware, we show the AMPA receptor recycling. So indeed, we still don't actually know what is late phase LTP. We do observe in the early phase phosphorylation of AMPA receptors, but that does not persist. Indeed, there appears to be more insertion of AMPA receptors into the spine. But that by itself is not sufficient. So you need slots. You need more PSD proteins. You need more of these other structural proteins. The spine head grows. We do know that you need actin reorganization, right? The actin has to unbundle to reorganize, and then it grows, and then it rebundles. So there's a lot of things going on. I didn't show you, we do indeed have phosphorylated AMPA receptors. Indeed, all the paradigms that produce this early phase LTP, including the one train of 100 Hz, which doesn't produce late phase, will produce an increase in AMPA receptor phosphorylation. We have not yet included AMPA receptor recycling, and so our AMPA receptor phosphorylation is transient and then goes away. But there's some other studies, I think out of Upi Bala's lab, which shows that if you then add the recycling, you can show that which is affected by the phosphorylation, you then have a more persistent AMPA receptor phosphorylation. But that's still not late phase LTP. So we want to direct our efforts toward the effect of these three molecules on actin. So now instead of having this molecular signature, we'll be able to use perhaps the state of actin as the tag. Right? So actin might have to unbundle to allow diffusion of these proteins that are synthesized into the spine. So unfortunately, our models can't keep up with the new experimental data, but we're always trying to add more aspects of what the experimentalists think is going on to be able to use their experiments, but then go further and ask questions that maybe haven't yet had a technique invented yet or something.
More questions? Yes, please.
Yeah, this is a really cool model, and it's really impressive what you were able to compute with it. I have a kind of mundane question, but I think it's important for the people that are like practicing this sort of thing. Like, how did you come about the parameterization of the model, like rate constants, and how is it? Like, is there any qualitative effect on the results if you fiddle with them?
Yeah, so that's a big headache for us. That's almost as big a headache as waiting for the simulations to finish. So we love the new cool imaging techniques. Particularly, we like FRET techniques, which show us. We also read the biochemical literature. So we use lots of different sources. So we'll look at kinase assays when we can. We talk to other experimentalists who might tell us, look at this person's data, not this person's data, because we know that not all data is equal. And then, so that's one source. It's much easier to get affinities and KDs than KCATs, and actual speeds, but sometimes you can get those. Then you have things like imaging of cyclic AMP or imaging of, in the striatum, DARP32. And so that's another constraint, like a constraint not on a single rate, but on short pathways that we then use to tune our model. So we have varying degrees of constraints. So I would say some rate constants are very well constrained and some not so much. I didn't show you here, but often when we're tuning these parameters, we'll either explicitly do some variation of our least constrained parameters to make sure that our main result is not contingent on that particular set of parameters. That's why we were very happy about this range. This doesn't show you here, but I won't go through it. But I said there was a range of thresholds. If there was a single precise threshold, I would not be real comfortable. In some cases, as we're developing the models, and then we might later get better parameter estimations, and we'll change things, and it's like, thank goodness we're getting qualitatively the same results. But we don't do formal parameter sensitivity analysis, in part because when a single run takes you a day or 3 days, it's not really practical. So that's why we do a more selective parameter sensitivity analysis. We're starting to get into parameter optimization and trying to figure out how we can do a limited amount of tuning, more automatic tuning with perhaps simplified models, but we're not there yet.
I think that concludes. Thank the speaker again.